Hello, we want to learn together the virus host interaction. Coexistence of viruses with their host can be viewed as a molecular arms race between the virus and host elimination mechanisms. Continuous interaction between hosts and pathogens during their coevolution have shaped the immune system, and in turn viruses have manipulated host immune control mechanisms to facilitate their propagation. Today, viruses are so small and simple they cannot even replicate own host on their viruses carry only the essential genetic information they need to be able to sleep inside a host cell and win over it into making new copies of the virus. The influenza virus, for instance, has only 11 protein coding genes because viruses are usually so basic. Many biologic many biologists didn't think they could even be classified as a life form. Basic information on understanding the name of the virus is presented on figure 1. Understanding the name of flu viruses. A. Sydney 0597 H3N2. The A is a virus type, the Sydney is a place virus isolated, the O5 is a strain number, the 97 is a year isolated, the virus subtype is a H3N2. How viruses are named? So many different types of viruses exist that nearly every living organism has its own set of viruses that try to infect its cell, even the smallest and simplest of cells. Prokaryotic bacteria may be attacked by specific types of viruses. Bacteriophages, see previous lecture, and I also explained it on the previous lecture video. To infect a host cell, the virus must first inject its own nucleic acid into the cell through the plasma membrane and, if present, the cell wall. The virus does so by either attaching to a receptor on the cell surface or by simple mechanical force. The virus then releases its genetic material, either single or double-stranded RNA or DNA, into the cell, and cells become infected. The virus nucleic acid uses the host cell's metabolic machinery to make large amounts of viral components. The virus's nucleic acid uses the host cell metabolic machinery to make large amounts of viral components. In the case of DNA viruses, the DNA transcribes itself into mRNA molecules that are then used to direct the cell's ribosomes. One of the first polypeptide to be translated destroy the host's DNA. In retrovirus, which inject an RNA strand, a unique enzyme called reverse transcriptase transcribes the viral RNA to DNA, which is then transcribed again into RNA. Once the viral DNA has taken control, It induces the host cell machinery to synthesize viral DNA protein and starts multiplying, as shown on figure 2. Okay, it's a figure 2 and it's about the HIV replication. You see, it's here the virus as a fusion, and we have a fusion inhibitors also. Reverse transcription of viral RNA genome happens. Reverse transcriptase inhibitor can comes here. Genomic RNA comes by the reverse transcription, the double stranded DNA, it's a product. And then we have a provided RNA come to the cell nucleus. We have a provided RNA. Integrase inhibitor can come here and inhibit the integration. Then transcription happens and the viral mRNA, viral protein, translation, and the cleavage of polypeptide and assembly can be happens by the protease inhibitor. Finally, the viral release and maturation of the HIV happens. Among on the surface we have a chemokine receptor CCRS or CXCR4, and the chemokine receptor antagonist can come here. We have a binding to receptor CCRS or CXCR4, an attachment to CD4 receptor. It's written here when it's coming, then the fusion happens. First attachment, then binding, finally fusion. About 25 minutes after final infection, about 200 new single individual particles of a virus, so-called virions, are formed. Once enough virion have matured and accumulated, a specialized viral protein are used to dissolve the bacterial cell wall. The cell bursts. That means it undergoes lysis due to cell wall due to high internal osmotic pressure, water pressure, then that can no longer be constrained by the cell wall. This release progeny virions into the surrounding environment, where they can go to infect other cells, and another lytic cycles begins on figure 3. Figure 3 is a lytic cycle. The first is the attachment of the bacteriophage. We have a host and bacterial cell attachment. The phage attaches to the surface of the host. Penetration. The viral DNA enters the host cell. Biosynthesis. The phage DNA replicate and phage protein and mates. Phage protein or mates. Maturation. New phage particles are assembled, and the cell lysis releasing, and the newly made phages. The phage that cause lysis of the host is called a lytic or virulent phages. Some lytic phages undergo a phenomenon known as a lysis inhibition, where completed phage progeny will not immediately lyse out of the cell if extracellular phage concentration are high. Lysis inhibition. Lysis from within lysis from within Li and lysis inhibition LIN release virion and secondarily adsorbing virion on the tail tube shown. Peptidoglycan, peptidoglycan cell wall, inner membrane and plasma membrane. In contrast, the lysogenic cycle does not result in immediate lysis of the host cell. Those phages able to undergo lysogeny are known as temperate phage. Their viral genome will integrate with host DNA and replicate along with, with it fairly harmlessly, or may even become established as a plasmid. The virus remains dormant until host conditions deteriorate, perhaps due to the depletion of nutrient. Then the endogenous phage, known as propophage, becomes active. Then the endogenous, known as propophage, becomes active. At this point, they initiate a reproductive cycle, resulting in lysis of the host cell. As the lysogenic cycle allows the host cell to continue to survive and reproduce, the virus is reproduced in all of the cell's offspring. An example of a bacteriophage to follow the lysogenic cycle and the lytic cycle is the phage lambda of Escherichia coli. Figure 5. Phage life cycles. Temperate phage can insert their genomes in red into host bacteria, chromosome in blue, to become a propophage in the lysogenic life cycle. Following lysogenic induction, propophage exists and enter the life cycles. Okay, the host bacteria, phage comes to the host bacteria, and then lysogenic pathway. We have an insertion of the plasmid to the DNA, host cell replication, because the bacteria has a binary division, and then lysogenic induction. Again, we have a lysogenic induction. We have two pathways here. Lytic pathway, phage assembly, and it's a short to comes to the phage release pathway, or lytic pathway, or lysogenic pathway. It's come here, host cell replication, and lysogenic induction. It's come out as I see. And then phage assembly. Following lysogenic induction. To become a prophage in the lysogenic life cycle following lysogenic induction. As a lysogenic cycle allows the host cell to continue to survive and reproduce, the virus is reproduced in all of the cells of spring. An example of bacteriophage known to follow the lysogenic cycle and the lytic cycle is the phage lambda of Escherichia coli. Viruses that infect plants or animal cells may also undergo infection, where they are not producing virions for long periods. An example is the animal herpes virus, including herpes simplex virus, which cause oral and genital herpes in humans. In a process called latency, these viruses can exist in nervous tissue for long periods of time without producing new virions, only to leave latency periodically and cause lesion in the skin where the virus replicates. Okay. Figure 6. Schematic representation of acute and latent herpes simplex virus. Acute infection, you see the virus is comes, the A, and the B, the DNA comes, there's the C, it's the viral genome comes to the DNA, alpha protein, beta protein, gamma protein. I don't understand what's happening here. And D, and then it comes out lysogenic, or that's a lytic pathway. It's written here latency associated transcript or LAT. And it's come to the cortex, hippocampus, in the trigeminal ganglion, and the cortex. Okay. Even though there are similarities between lysogeny and latency, the term lysogenic cycle is usually reserved to describe bacteriophage. Lysogenic cycle is usually reserved to describe bacteriophages. A special cell of the immune system called the T cell circulates between the blood and the lymph. Looking for infectious agents, cytotoxic T cells have a specialized protein on their surface that helped them to recognize virally infected cells. These proteins are called T cell receptors. Each cytotoxic T cell has a receptor that can specifically recognize a particular antigen peptide bound to an MHC molecule, part of a special major histocompatibility complex, which is a set of genes that code for a cell surface protein essential for the acquired immune system to recognize foreign molecules. Okay, even though there are similarities between light and T and a special cell of the immune system called T cell, Cytotoxic T cell have a special protein and this effect and not tells the human protein by this. I hope I learn. I hope. Especially thanks for Dr. Ayjan Jospova, Dr. Professor Azad from the Polytech also.